Okay, so I think if you remember in the last class, we looked at JPEG DNA standard. I think obviously there are many more possibilities in that area. Now, today I'm going to look at DNA cryptography. Okay, so because if you remember, the title of the course is DNA storage and security. Security part we should start. There are about 13 lectures right now, and towards the last one or two lectures, I will cover main techniques about synthesis and sequencing because that is also important, DNA writing and reading. So, but I think in the next couple of lectures, you will see the flavor of cryptography, DNA cryptography, right? So, okay, so this is lecture 22. Now, so this is the picture, if you remember, is the how DNA storage works. By the way, this is, we are talking about, there are two kind of DNA storage. I think till now we have discussed about sequence specific storage, right? So you convert data into the DNA sequences and store into in the form of dry DNA or liquid DNA or whatever. But there is another approach which is quite popular these days coming up, which is known as the structure based DNA storage. So on the structure of DNA, they store information. Okay, so that is another beautiful area. And in fact, one of them is also involving cryptography in the structure. So we'll talk about that also. So first, let me focus on the DNA cryptography. So this is the standard picture, as you know: encoding, decoding, storage, synthesis, retrieval. Now, I think some of you might have done a course SC402 cryptography. How many of you have done it? So almost half of them. Fine. So cryptography is very important. I cannot cover the whole cryptography course here, but I'll just few basic things, and you can look at the videos of cryptography, which is available anyway. So Bruce Schneier is a very famous cryptographer. It's a very famous book, Applied Cryptography. If you want, you can start from this book if you want to learn cryptography. And he has said a statement which I like, that there are two kinds of cryptography in this world: cryptography that will stop your kid sister from reading your files, and cryptography that will stop major government from reading your files. So now, in terms of DNA storage and security, the last statement is again very, very important. Uh, in fact, I could not touch a very important topic in cryptography course last year, which was fully homomorphic encryption. It's a very, very beautiful technique. And the idea to fully homomorphic encryption actually was due to the same people who invented RSA, right? Rivest, Shamir, and Edelman. So Rivest and Edelman, and there was one more scientist. So when they were looking at RSA, so RSA crypto system has a very peculiar property. So what happens? You encode the things, and then the encoded things, if you multiply, so you can actually find out what is the actual multiplication of the things which you encoded, right? So you understand what I'm saying? So there are two things. You do the encoding and then you multiply them. So you can do the operation on the encoded stuff. And actually operation you can happen on the actual stuff, which was there. This is because of a mapping which is known as homomorphism. That's why it's known as fully homomorphic encryption, right? Which is preserving the structure. Okay. And this is a very beautiful technique. So what are there are wider applications of this technology? For example, you know that all these days we store most of the data into the cloud, right? And we don't want to actually give us our information about ourselves, right? Our photographs or whatever, or maybe financial information, right, to the service provider company. But at the same time, you want that service provider company should be able to suggest you, for example, YouTube, right? So you should know that YouTube should be able to suggest you the recommended videos. How they will suggest, unless they know your interest and your basically, location and other things, right? Now, with fully homomorphic encryption, all this can be done and the data will be encrypted in the servers, right? So basically, actual company will not know what is a thing, but the algorithms will be knowing everything and they are doing the calculation on the encrypted data. It's really very, very powerful technique. And this technique is going to be quite useful for DNA storage actually, because this is something, which you can look at it, right? So fully homomorphic encryption is very, very common thing. There are challenges also, especially in the terms of biology, to provide privacy to the genomes and so on. Anyway, so that is something really interesting. So as I mentioned, if you have not done the course on cryptography, it will be nice idea to look at these 23 videos, of cryptography course. You can have, it's about 23 hours. Maybe each hour if you look at in the fast mode, maybe 15-20 minutes, right? So you have less input time to understand what is going on anyway. Now, so cryptography, so there are three kind of terminology. Cryptography, the study of building secure crypto system, right? Then second, cryptanalysis, the study of breaking crypto systems. This is very, very important. And cryptology, the study of both cryptography and cryptanalysis. That is what is the branch is known as cryptology, right? So these are the three basic things. I'm just recalling from the last year course, few basic things about cryptography. Then there are four kind of things that you look at into cryptography. First is classical crypto system like ciphers. There are different kind of ciphers, right? Substitution ciphers and so on that you look at. The classical crypto, that comes under the purview of classical cryptography. Then we have private key cryptography, symmetric cryptography. All these block ciphers, for example, DES, you might remember. DES, AES, these are all block ciphers, okay? So they are basically private key cryptography. Then we also have public key cryptography, like RSA is one such example, which is in the public key cryptography, or electrical crypto system is also in a public key crypto system. And then of course the protocols, how do you implement and then do all the business, right? So these four basic areas. Now in terms of DNA cryptography, some work has been done in the area of classical cryptography, some work has been done on private cryptography, and some work has been done on a public key cryptography. And apart from that, there are many new ways of looking at information security in a totally different way because of biology is involved, right? So this is something, very, very important. Okay, so I'll give you brief idea because I think probably I don't think there is any course on DNA cryptography anywhere, right? So probably here you will see what are the things available, right? Now, so I'm just giving this again, recalling all these facts because so that now when we look at biology, you can see what has been done and what has not been done, right? So we know that there are different type of adversaries that are possible. Alice is sending something, Y, to Bob. Bob should receive Y, but adversary is stealing that information, right? That is one kind of passive adversary, they call it, right? Then there is another one you send here. Alice is sending Y, and adversary is sending Y prime rather than Y to Bob, right? That's another problem. Alice is not sending anything and Bob is receiving something, right? That's another problem. Or Alice is sending something and rather than Bob should receive, Charlie is receiving Y. Someone else is receiving Y, right? So that's also another problem, right? So these are the different kind of adversaries that are possible. I think these are some terms: message integrity, message authentication codes, cryptography protocols, Kirchhoff principles, signature schemes, certificates, hash functions, attack models. Very common known terms which you should recall from cryptography course before we look at into. So I'm not covering anything in details, right? You can look at the cryptography course for this. Attack model, of course, I would like to again review briefly. So there is a ciphertext only attack. So opponent possesses a string of ciphertext Y. So basically, normally in cryptography, there is a string X, which is plain text, and a string Y, which is ciphertext, right? Encryption of that, right? So if the opponent possesses a string Y, a string of a ciphertext Y, then you want to decrypt. That is one. So this is known as ciphertext only attack. Known plain text attack. So opponent possesses a string of a plain text and the corresponding ciphertext, and then you find out how things are happening, right? At least if you can, from this pair, you can tell information about other pairs, right? Chosen plain text attack. The opponent has obtained temporary access to the encryption machinery. Hence, he can choose a plain text string X and construct the corresponding ciphertext string Y. That is chosen plain text attack. In chosen ciphertext attack, opponent has obtained temporary access to the decryption machinery. Hence, he can choose a ciphertext string Y and then construct the corresponding plain text string X. So all these 4 kind of attacks are there, right? So now depending upon the biological system, you will see that how beautifully people have used different kind of techniques in biology, especially when you mix cryptography with biology. And of course, you can recall what is computational security, probable security, and unconditional security, right? What are these important ideas? You can recall from the cryptography course. Then I think in general, there are 2 kind of cryptography: key-based, keyless, like Caesar cipher and so on. Then asymmetric key, symmetric key, protocol level, and then also you have basically RSA and other things are asymmetric key. Block cipher, symmetric key. There are DES, okay, digital encryption is standard, or data encryption is standard. 3DS, triple DES, blowfish, AES, advanced encryption is standard, IC and others. RC4, RC5, those are the techniques in the stream ciphers. Okay, so reverse scheme, so there are those kind of schemes, and in fact, year-wise they are given, right? RC2, RC5, RC6, AES in the block cipher and the stream ciphers, right? And the asymmetric we have RSA. This picture captures most of the commonly used systems, right, in the cryptography. So if you have some of these ideas, actually you can now see what happens in biology when we mix these things into the biology. For example, I will show you what happened, the first experiment of DNA computing was done in 1994. Immediately in 1995 and 96, people attacked DES using those techniques. So, so how biology or how the DNA can be used to break those techniques, that was shown actually. It's a really beautiful paper. I'll show you those papers also. Okay. So I call it as a next generation cryptography, means where cryptography is coming to a next level. Of course, quantum cryptography could be part of it, which I have not included here, right? And post-quantum cryptography system is also part of this quantum cryptography, post-quantum crypto system, right? All these are part of next generation cryptography, but I am focusing on biological. So it is also known as biomolecular cryptography. Okay, biomolecular cryptography. Of course, this list could be very long, but I have kept few of them. Classical DNA cryptography, so which is started early 95, 96, and so on, so forth. Dry and wet DNA security. So once you convert data into the DNA, either liquid or dry, you also require security of that data, right? So that's a direct challenge. Then DNA origami cryptography, I'll tell you about DNA origami cryptography because some work has been done there. It's a nice, beautiful work. And of course, there's a lot of possibilities here, what can be done in future. So I'm going to discuss that in the next maybe 2-3 class from now. JPEG DNA security. As you know, the JPEG DNA standard is developing, and they have observed that JPEG DNA security is a critical component, but not much work has been done on that. Still it is quite open. Blockchain and DNA storage has a very interesting relationship. So blockchain, you all know, and what are the possibilities? So sometime I will discuss in future what are the possibilities of blockchain with DNA storage. Privacy of genomes. This is something very interesting. So you know that you have your, this your DNA, right? Your DNA has the entire blueprint of making who you are, right? Your eyes are brown, black, or all kind of things, all the shape of your nose and everything is encoded into the DNA, right? Now, companies are there. For example, in India, there is a company Genome Patri. In US, there is a company 23andMe, which are actually taking the sample of DNA and doing different analysis. For example, predicting what kind of diseases are possible to you, right? Now, but once they have your DNA, that means they have the entire picture of you. I mean, everything about, they know everything about you, right? So in that sense, how do you provide privacy there? That's a challenge, right? You want to encode the genome uh, in such a way so that privacy is there. So there is a competition iDash. I don't know whether you heard competition iDash where they are challenging cryptographers to provide best cryptographic mechanisms so that you can secure the genome actually. And I think, as I mentioned, fully homomorphic encryption technique, which was, of course, suggested in 70s, but finally it was discovered in 2009. Okay, and then just after this discovery, there is a Turing Award winner scientist. He said that this will be of no use because it is very slow. The whole process of fully homomorphic encryption is very slow, so probably we will not find any practical utility of this, although it's beautiful. But it turns out that right now you can actually secure the whole genome with in a couple of hours actually. So right now people are, so fully homomorphic encryption has evolved at such a level, right? So this is something which is very, very important. From biology point of view, fully homomorphic encryption is very, very important to provide privacy. Okay, so this is something very, very important. So obviously you will find many interesting problems to work on. The whole field is unexplored much. Okay. Lastly, this is also a very dangerous thing. DNA malware cyber attack. So what happened here? They took a virus, computer virus, converted into the DNA, right? So that exe file virus is converted into DNA, and that DNA is shipped. So suppose you receive DNA, you attach that DNA to your machine, which is DNA sequencer, and the DNA sequencer will be connected to a desktop or your machine computer system. And so that DNA string was converted into again a exe file and which has infected the system. So malware attack on the machine is possible, that they have shown. So it's known as DNA malware attack to the cyber attack basically. You send a DNA to someone, they are synthesizing and then they are they are doing the reading it and then it is executed. So those kind of things are possible. So basically you can, and this is very, I mean, this conversion is very useful also. Suppose on Mars, you want to produce different kind of viruses or bacteria. You take the genome, right? Only thing you need to synthesize those things there, and genome you can send it, it's a digital file, right? Transmit bit by bit, somebody will receive it there, and you execute. So all kind of possibilities are there, right? So anyway, this is something really interesting. So they have done successfully in 2017, if I remember the correct year. Okay. So at each of these places, plenty of opportunities are available for you to do something in DNA cryptography. Okay. Now, in order to tell you more about DNA cryptography, I need to give you some background about DNA computing as well. I'm not going into too many details. Again, if you want to know more about DNA computing, we have a full-fledged course available on YouTube. You can watch Natural Computing, right, which talks about in details about DNA computing. Okay, so let me start. So what happened? If you remember your discrete math class, I think I might have taught you discrete math, right? So there I talked about those four kind of machines. What about the four kind of machines? Any computer scientist should know. Finite automata, then push down automata, then Turing machine, and there was another one in the middle. Total four type of machines were there, right? Finite automata, DFA, NFA, all those things. Then push down automata, which has one stack memory. Then linear bounded automata, and then Turing machine. All these four machines were there, right? So if you want to basically show that, so now we have understanding of these concepts from a computer science perspective, and we have the biological construct in front of us. I want to make a computer out of DNA. What I should be able to do? I take some DNA molecules, and I show that you can do any of these four computations with the help of DNA. Then we are done. And that was actually done by Eric Winfrey. Okay. So in 1997, 95, 97 actually. Though he submitted his thesis in 98 actually. Yeah. So this is something really interesting, that we should do this kind of matching. The first work in this direction was, I would say, that was Thomas Head. Thomas Head. What he did, if you remember central dogma from our previous lectures, DNA to RNA, RNA to protein, right? In between, there was another operation that I have not told you, which is known as splicing operation. DNA splicing operation. So what happens? Whole DNA is there. You read portion of DNA from one place, another portion of DNA from other place, another portion of DNA from other place, and then combine everything together to form a single mRNA sequence, right? And then that mRNA will convert into the DNA, right? To the protein, basically. So what happens? There is a splicing operation that happens. Now, Thomas Head actually has shown, he was a purely mathematical person. He has actually shown that this is equivalent to Turing machine, splicing operation on DNA. And this paper, an analysis of the generative capacity of specific recombinant behaviors, purely theoretical, 1987, formal language theory and DNA, right? Because this classification is known as formal language theory classification, and actually you can map it to the DNA, right? So this is the first way, purely theoretical way. So he looked at the splicing operation and how you can construct a Turing machine kind of thing from there, right? That was the thing that he has done in the paper. As you know, if you do theory, it will remain theory, nobody is interested, right? Because money will not come, right? So then what happened? Rivest. So in the Rivest, Shamir Edelman, RSA people, the Edelman was the person who was, I still remember I met Edelman in, I think, 2005. 2005, I met him. By the way, we have a lot of communications earlier on the email. Anyway, so Edelman was actually, what he was doing, he was reading a book by, that Watson and Crick, the book actually, molecular biology. So he was reading that book, and when he realized all this central dogma and other things, reading those things, he realized that these things can compute probably. I can make a computer out of these things. And but then he should be able to solve a problem. And that too experimentally, right? Then only you will say that it's an important thing, right? Theory is fine. So what he did, actually, he in fact told to his wife Laura that these things can compute. And next day he planned an experiment at University of Southern California. He went to his friend who was doing biology, the lab. Within 7 days, he learned all the biological lab techniques and performed the experiment. Did the experiment, he solved Hamiltonian path problem using bunch of DNA strings, and that was a seminal paper at Science magazine. So important discovery of DNA computing. Okay, right. So he solved instant of Hamilton path problem. You know that Hamilton path problem is quite difficult to solve, right? So just one instant using DNA strings. So people started thinking maybe DNA computing is a threat to complexity. Maybe you can solve many more complicated problems using that. But of course, it's an instant of the problem, right? We know that maybe in the way that how people are looking at DNA computing may not be able to help you to solve the complexity of many problems, but there are different ways of doing certain things. Of course, there are plus, minus, both. Anyway, so that is the thing. So this was the first experiment in 1994. See, this year is very important. Why 1994 is very important? It's the birth of DNA computing. And also, do you remember anything happened in quantum computing in 1994? It's very, very important. 1994, Peter Shor discovered polynomial time algorithm for factoring an integer and discrete log problem that you use in cryptography. So actually he hit the nail on the bottom of both the crypto systems, which were based on, so RSA and elliptical crypto system, both were based on difficult problems, right? Like discrete log problem is a difficult problem, and similarly integer factorization is a difficult problem. And those two basically problems can be solved in polynomial time on a quantum computer that he has shown in 1994. So it's a great discovery. That means all your crypto systems will be of no use if you have a quantum computer. And that's how the whole, money started flowing into the area of quantum computing. All the governments were interested and so on. Because it's a threat to the crypto system, right? Everybody is interested in quantum computing. As you know that now, if you go to IBM website looking for internship, if you have IBM Qiskit program you have done, probably it's easy, you can get easily an internship at IBM. So a lot of many companies are working, IBM, Google, Amazon, right? D-Wave, there are many companies actually, around 55 or so, as far as I remember. Anyway, so that is something. Okay, so this was the year 1994. So what is DNA computing? So this course, what we are doing is DNA storage. Essentially, before that, there's a lot more things that has happened. So basically, I would say that DNA computing has evolved in many directions, and I was also doing a lot of other things. And suddenly, this application of storage has come, which is really beautiful, and the people started focusing more on storage than the other things. I have also done the same thing. So basically, we also started focusing on DNA storage. So essentially, how it works. So in a normal computing scenario, we know that there is a computational problem, there is a computer, basically there are a bunch of circuits doing binary operations, and the output is in the form of binary strings, right? Okay. So any computational problem, you can break into these binary strings and you do the computations. In case of DNA computing, we have computational problem, and you convert everything into the DNA strings, ACGT. Now, as you know, in DNA 101 course, I have told you earlier also, A and T are complementary and C and G are complementary, right? There are different hydrogen bonds between them, right? So whenever two strands of ACGT are there, and if they are complementary will come into picture, so they will join. Actually, Edelman thought a very clever way of combining the strings. So first, he encoded each station of the Hamiltonian path, each vertex as a bunch of DNA strings, and the path also with the help of DNA strings. Now, these strings you put into the test tube, they automatically self-assemble because of the hybridization property, and you do the computation for you. And the output is you can read actually. Okay, this is something really beautiful. Okay, so everything is in the test tube, DNA operations, and output is also in the form of DNA string. Essentially, you need to find a way to convert any mathematical or computational problem into the bunch of DNA strings. Okay, and then you proceed. Now, why I am showing you this application chart here. So we have different kind of ways people have looked at it, emerging areas. One is DNA tiles. I'm going to give you some idea about it because in the first crypto system, they've used tile system. I'll show you how they have done that. DNA nanostructures, that is very, very important. Actually, I would say that this DNA cryptography can be done in all these components, 1, 2, 3, 4, 5, 6. All these, 1, 2, 3, 4, 5. All these 5 system will be a subset of DNA cryptography. You can do cryptography with any of these things. So what are these things? Let me tell you. DNA ties. So there are certain ties which can combine itself together to do the computation. This idea was there with S1 in 1961. Now, what happens? Eric Winfrey mapped each of these ties into the DNA molecules. There is a specific kind of molecules and which can do computation for you. So each tile corresponds to a DNA molecule in terms of hardware. Okay, so that is something interesting thing. So that's why now we have the abstract tile assembly system. I will talk about it in a minute. So that abstract tile assembly system can solve any computational problem. So basically, if you want to be a DNA programmer, you need to have a programming skills. How do you program a DNA computer? So if you want to program a DNA computer, you should know how to write tile system for any computational problem. Okay. So I think we have, in our papers, we have produced tile system for computing real numbers. Okay. A lot of things. You can do a FFT, fast Fourier transform using tile system. You can do discrete cosine transform, anything you want. Actually, so you have to produce a tile system which will do the computation for you. That is DNA tiles. Now, some of these things, this is a beautiful area, DNA strand displacement. In fact, there is a storage system also based on DNA strand displacement, DNA storage system. So what happens, DNA is, it's something you can think like this. There are a bunch of DNA strands fighting with each other, fighting with each other and doing computation. So you give a input to these bunch of DNA strings which are fighting with each other, or I would say the dancing. They are just dancing and doing computation and output is there, So this is known as dynamic strand displacement, of the strings. And you can actually build a storage system also out of it, which I'll talk about it later on. But this is something really very, very interesting thing, right? So why this area is important? As you know, in computer science or in electronics, I would say, any bigger circuit can be composed of some basic logic gates, right? There is a decomposition theorem, right? So you can actually build everything, anything complicated can be done from the basic logic gates, right? So basically, similarly, in chemistry, any chemical reactions can be broken down into certain basic chemical reactions. Okay, so beautiful analogy you see. Now that means, so Eric Winfrey has again shown in 2007 that bunch of chemical reactions can implement any machine. So you can implement Turing machine using bunch of chemical reactions. So just chemical reactions can do computation for you. So that means entire chemistry is computer science. And on the other hand, inside our body, there are a bunch of reactions happening all the time. So there are different computation machines implemented from the hardware side is chemistry, right? Inside our body. So computer science is everywhere inside our body. This is a beautiful connection between biology and computer science, right? That is what here. Now, you can also make different kind of circuits. Actually, there are on DNA circuits, I should say that there are two kind of things. There are scientists who are studying circuits inside the biology. Means if I look at human or Drosophila fly or any sea urchin, for example, sea urchin gene 16, they try to map the entire circuit. So there are biological circuits at the back end for any computation. So that's laying down the entire diagram of that circuit is a complicated thing. So I think if you so they have done it in components, for different species. For example, endo-16 gene of, endo-16 gene of sea urchin, and similarly Drosophila fly, that, right? Fly, they have studied the circuits. So if you actually, for one organism or a species, you can give the complete description of a circuit, you will get Nobel Prize, because it's such an important thing. You are actually describing the entire computational details of that organism at the ground level in terms of electronics, right? I mean, rather than saying electronics, I should say circuits only, right? Because it may not be in the form of electrons. But anyway, so that is one aspect of DNA circuits. Secondly, using any of these things, for example, DNA strain displacement. So these DSD, it is short form is DSD, dynamic strain displacement, can implement any circuit. You can have NOT gate, AND gate, different kind of gates using bunch of DNA. So DNA strings dancing together and doing the circuit implementation is possible actually. So sometime I can give you those things. By the way, in the natural computing course, I have talked about some of these things. There are about 7 lectures on chemical computing. Chemical computing means computer science, chemistry looking at as a computer science. Okay, that is this thing. Now DNA storage system, as I mentioned, right now we are looking at sequence-based storage, but you can also look at structure-based storage. And there, even this portion is coming into picture. Apart from that, there is a NIC-based storage that people are using. Okay. So now, DNA origami, so DNA nanostructures, you can encode information. See, these viruses are very beautiful. COVID-19 is a very beautiful virus, right? Mathematically. You'll see beautiful symmetry. You look at any virus, RNA viruses are beautiful mathematically. There are nice, lot of interesting, results, and also connections with group theory, for example, all these viruses. Okay. So that is there. So in structures, you can also do cryptography with the structures. So that is what I will show you. One of them is DNA origami cryptography that I talked about. And of course, so DNA storage, sequence-based storage, and structure-based storage. So that means sequence-based cryptography and structure-based cryptography. There are two kinds of cryptography in the DNA. Sequence-based DNA cryptography and structure-based DNA cryptography. शायद मैं ही पहली बार term introduce कर रहा हूँ, right? But that's okay. So we are introducing these terms. These are, nice thing to look at. Right. So this is all. I think I hope you understood about all of this application. So basically, you can make crypto system with any of these things. Entire things are open. Okay. So now, after the first experiment was done by Eric Winfrey, sorry, not Eric Winfrey, so Edelman in 1994, immediately scientists started jumping, oh, wow, this is a new thing. And then Lipton came into picture, Professor Lipton, Richard J. Lipton, and many others. They started building a structure or algorithmic structure of doing computations. So, for example, in the form of test tubes. Most of these methods involve what is known as filtering methods. So basically, you encode the problem into the DNA strings, hybridization will happen, some output will be there, and you filter it out, the solution out of it. So there are different kind of filtering mechanisms exist. There are at least 7 or 8 filtering mechanisms exist right now, just with the help of test tubes. So there is a new kind of data structure, I would say, in terms of DNA computing is there. But actually, this is all, it's not autonomous. It's all non-autonomous. A lot of manual things are involved, right? So obviously, not much progress will happen. Of course, there were some initial papers, but basically, using these filtering models, initially, what people did, immediately they attacked DES with the DNA computing, and then see if we can get some success. And actually, they did get some success in breaking the DES, I forget the number of bits actually, but probably in the next slides, I will show you how many bits they actually were able to break using this. So DES was able to, so DES, right? So data encryption is standard, right? How you can, so it works on Fistel network, if you remember. Block cipher, 64 bits going out, in, and 64 bits going out, and some sort of round functions at each step, if you remember. DES from the last lecture. So that is something. So after that, Eric Winfrey, who is now at Caltech, so he said that maybe we should automate this whole process of DNA computing. So the idea wise, it is fine, Edelman has given, but we should automate. So he took the idea, the three things, Wang tiles, S. Wang in 1961. I'll show you what is this Wang tile business and how it solves certain problems, okay? And the second thing is hardware. Ned Seaman actually died very recently, this year only. Ned Seaman, I also met him in 2005 only, So I know, so some of his friends are good, some of his students are good friend of mine actually still. But so Ned Seaman, what he used to do, he did by trial and error, he made cube out of DNA or some other structure, mathematical structure out of DNA by trial and error, no algorithms, nothing. So he has created a lot of library. What is known as some of the molecules are double crossover molecules, DX molecules, triple crossover molecules, TX molecules, made out of DNA based on hybridization technique. Okay, so this was done by Ned Seaman. So Eric Winfrey took the initial idea of DNA computing from Edelman, took the algorithms from Wang tiling business, and took the hardware from Ned Seaman's lab and created what is known as algorithmic self-assembly. So DNA hybridization is a self-assembly process, and can we create an algorithmic version of it so that we have full control what we want? And that's what actually Winfrey was able to do. So you can look at his thesis. It's a beautiful thesis. It's still available on his homepage, okay, where he talks about how you can do different things. So this is the first idea of Wang tiles. You look at these tiles. So you can see that each tile has different colors at each edge. So yellow, red, yellow, yellow, or red, green, green, blue, the first tile. Second tile, is red, green, blue, green, like that. You have all the different colors tiles. How computation can be done? We'll say that. So basically, we can define certain operations on these tiles. Two tiles can come together and join each other if their corresponding edge color matches. So for example, if this, if the first tile and the second tile come closer, then they will not match because the green and red will not match. So they will not join. Okay. Whereas the second row, if you look at this right side color of the first tile and left side color of the second tile, both are yellow, they can come together and join. Okay. So joining mechanism is if the color matches of the corresponding thing. Of course, there are assumptions that you cannot rotate the tiles, otherwise there will be a problem. Rotation is not allowed, right? You need to be in one framework, right? Then this kind of coming together can do any computation. You can build Turing machine out of it. That's a beautiful thing. That was shown by S. Wang in 1961. Means with these tiles, you can do computations, right? So this is a beautiful idea, right? So these are known as Wang tiles. I think probably I will stop then. In the next class, we are going to talk about a few more basics, and then we'll go to the DNA crypto system, how they are implemented, right?